recognized into lymphatic follicles or that we find in the spleen. And we'll look at them in more detail when we talk about the T cells and the B cells. But over here is the T killer lymphocyte. There are actually several types of T lymphocytes, and it has a direct interaction with whatever cell it's attacking. So T lymphocytes, T killer lymphocytes do not make antibodies. They will release chemicals, and there's a direct binding between the cell that they're attacking and the cell, the T cell itself. B lymphocytes, again, as a lymphocyte in the blood, you cannot tell the difference between the T and the B, just that it's a lymphocyte. Once the B lymphocyte comes into contact and its receptor recognizes the antigen for which it is specific, it converts to a plasma cell. We talked about this a little bit on Friday last week, Thursday and Friday. And so here we have the light area. Remember, the B lymphocytes feel like Woolsey and ribosomal ER, and that allows them to produce the proteins, which are antibodies. So once they're activated, B lymphocytes can be distinguished. And we'll talk about that clear area a little bit in a few minutes.
How are they activated?
We'll talk about this on Thursday and Friday, but they're activated when they have receptors on their surface that look like the specific part of an, in fact, they are antibodies. So they have these receptors, and they will meet up with a macrophage or a T cell that is presenting an antigen that matches that. Remember the antigen presenting cell concept we talked about? And if that matches that and binds to it, that triggers the cell to start making the antibodies that match, that look just like those receptors. And so they'll start making antibodies whose surface recognizes that same component that the B cell receptor matched. And so in the conversion to make those antibodies, they have all the Golgi and the ribosomal ER. To assist this, we have the macrophages. And remember, the macrophages in the blood are not activated except in the liver, and they are known as monocytes in the blood in their non-phagocytic form. And they have multiple long processes. These are the cells you learned about in Bio430 when you talked about the concept of endocytosis or phagocytosis. And a bacterium or a virus or a foreign cell, these long processes extend out through a amoeboid movement, fuse, and now that foreign pathogen is within a vesicle, interacting with lysosomes. Does anybody remember what reticular cells are? Reticular cells we first discussed in Bio430 when we were talking about different types of fibrous connective tissue. They're found in the spleen, in the lymph nodes, in the tonsils. They're really, let me turn the lights down here. These would be lymphocytes here, and this is the reticular cell here. It's a fibroblast that wraps itself around the collagen it makes. And so it creates this network of fibers that span connective tissue, and we find them in lymphoid organs like the spleen or tonsils. And they form a sponge-like network that stabilizes the lymphocytes. I told my students last semester, it's like trying to, you can't make a 10-foot pile of marbles. They're just going to roll away. And so this helps to entrap and hold the lymphocytes in a highly concentrated area. It also slows down the flow of fluid. So if we have lymphatic fluid percolating through a lymph node, it has to flow over and around the cells that are viruses or bacteria that are in that lymphatic fluid, have to bump into these fibers, and it slows them down, gives them longer time to be in contact with the lymphocytes. Any of them, the macrophages and the B and the T cells. But the round cells that you see here are the lymphocytes. And this would be a fibroblast wrapped around collagen fibers, and that's the reticular cell. A different type of a fibroblast.
So you can't tell the difference between the T cells?
No, not in this view. Now this one is really dark. It looks like it's already a plasma cell. So in that case, it would be B. But when they're in their round circular form,
No way to tell.
you can't tell. Now when we get to lymphatic tissue, B cells are more likely to be found in certain locations, and T cells in other locations. But by appearance, you can't tell. So these are the organization of the first part of the lymphatic. Let me get a different view here. So we're going to look at lymphatic tissue, and then we're going to look at lymphatic organs. All right. So as a tissue, we have diffuse lymphatic tissue, and then dense lymphatic tissue in which the cells, lymphocytes, are in a dense congregation. Then we take the lymphatic follicles and we put capsules or partial capsules around them, and that becomes our lymphatic organs, such as a node or a tonsil or the spleen. So that's the sequence of organization, very loosely organized, more organized, and then within a capsule is the progression in which we'll talk about them. Let me come back to the bottom part of that tissue. I want to kind of show you what I'm talking about here. So this is looking at the lining of the small intestine. And the surface mucosal membrane of the small intestine is thrown into folds. I have a model of those folds here. It functions to increase surface area. So these are villi, these are not microvilli. Each of these cells here is a simple columnar cell, and they will have the microvilli. This is loose connective tissue in here. And so we would find a high concentration, but scattered. All these little purple dots are primarily lymphocytes. So there's a high number of them, but they're not organized into a sphere, into a this form like you see here. And that's what we just call diffuse lymphatic tissue. Okay? So highly concentrated, but still spread apart. So you can see that's more concentrated than down here. So we have our diffuse lymphatic tissue. By the way, while I have this model up here, remember we talked about the beginning of our lymphatic tissue fluid flow was the connective tissue entering into lymphatic capillaries. And unlike blood capillaries, which were coming from an artery and going to a vein, arterial or veneal, the lymphatic capillaries were blind ending. And we have a special name for the lymphatic capillary in the small intestine, and that's a lacteal. So this is an illustration of a lacteal. And it picks up in the small intestine not only connective tissue fluid, but remember that's where our large fat molecules covered by proteins, coated with proteins, are going to enter into.
Can you spell that?
Lacteal, L-A-C-T-E-A-L. So if you were to identify that as a lymphatic capillary, that would not be wrong. You would get partial credit for it. A more specific term would be lacteal. So this is just a higher mag of diffuse lymphatic tissue. And you can see the scattered lymphocytes.
I promise I took an allergical test.
Now,
Waiting for it to kick in.
within diffuse lymphatic tissue, we never see this next structure without the diffuse lymphatic tissue being present. We have a lymphatic follicle. So all around here, you can still see this is actually in the colon, the large intestine. And we have diffuse lymphatic tissue up here, but now we have this sphere. All right, so it's shown in this model as this structure right here. Okay, where the lymphocytes are packed closely together. More reticular fibers would be present than in the diffuse lymphatic tissue. And this is organized such that the periphery of the lymphatic follicle is T cells and the center is B cells. So again, unless the B cells are activated, you wouldn't be able to tell by looking at them, but antibody tests would indicate that. So in the periphery, we have the T cells, and I know I'm drawing lines, but I'm not going to draw 2,000 little circles. So these are T cells.
say that back?
These are T cells, T lymphocytes
So you're gonna put the on the outside?
on the outside, on the periphery, and B lymphocytes would be in the center. And if you look at that, it seems like there's a lighter area, so they're not as dense. B cells are in the center, T cells are in the periphery.
Oh my God.
So this seems to look a little bit different here. There seems to be slightly more clear space around the blue dots than out here. And the reason for that is not always present. But if the B lymphocytes are activated, remember they're going to become plasma cells. And when a B lymphocyte becomes a plasma cell, it goes from this appearance to this appearance. And we have a light Golgi area and ribosomal ER, but that's less dense than the nucleus. All right, so let me take the slide back to the lymphocyte.
The plasma cell, right?
So with this is still lighter than the nucleus, and so it opens up the area of the lymphatic follicle, and it stains more lightly. So on these here, you can see that these centers have a lighter area, and that is identified, that lighter area is identified as a germinal center. And it's due to the B lymphocytes being activated the plasma cells. So it means that some of those cells came in contact with an antigen for which they were specific, underwent mitosis, made more of them, converted to plasma cells, and so that center is showing up lighter with the excess cytoplasm. So it just means we have active antibody formation. Back to the terminology. I gave you a choice of lymphatic follicles or nodule. I prefer follicle. You can use either or, and you will see either or. The reason I prefer follicle is because the first organ we're going to describe is the lymphatic node. And nodes and nodules get mixed up. Follicle and node, less likely. In fact, the lab manual that I use at Sac State, written by one of the faculty there, put in lymphatic nodule when it meant node. And I was just like, okay, just eliminate that word, and you're less likely to get it confused. Okay? So lymph nodes have capsules. They're accumulation of many lymphatic follicles. So this is a lymphatic follicle. They made it wrong. They should have made the center, the light blue, and the outside, the dark blue, to match the appearance of our germinal center, but that would be where the B lymphocytes are located. So we can find them as a single structure, similar to this image here, or we can find them in high densities in the appendix, or this is in the ileum. And the ileum is the last third of the small intestine. So it's right just before the large intestine. And normal flora of the large intestine are E. coli. We don't normally have that in the small intestine. And so if bacteria from the large intestine migrate into the small intestine, they can more easily get across the epithelial lining and into the body tissue. And so to protect that, if that happens, we have a lot of both diffuse and lymphatic follicles. Okay. So back to that first slide talking about MALT. The combination of diffuse and lymphatic follicles forms this mucosa-associated lymphatic tissue. So it's the combination of the diffuse and follicles, and we find them just under the epithelium of some of our organs that are exposed to the external environment. you look on the diagrams on page 46, there are two diagrams that are shown there on the left-hand side. One is labeled they both have the word capsule and diffuse lymphatic tissue. That's there just to show the lymphocyte, the scattered lymphocytes and the lymphatic follicle, but we don't have any capsule with these tissues. When we put a capsule, then it's in the lymph node, okay? So that was just a picture to show you what a lymphatic follicle looked like. Right. So when we find just a lymphatic follicle, it's not going to have any capsule associated with it. I don't want you to confuse it from that diagram. So you cross out the capsule words. So the capsule and trabeculae, cross out those terms.
What keeps you grouped up like that?
The reticular fibers.
Fibers and lumps.
So here's a lymphatic follicle. It has connective tissue as part of the submucosa, but not forming a dense capsule around it. Okay? So if something were, some pathogen were to diffuse through this epithelium, it would have to go all the way through all of these lymphocytes. It would then get into the looser connective tissue, or the dense irregular, and would get into the lymphatic capillary at that point, if it wasn't already destroyed. Now let's take these follicles, all right, and we're going to look at lymphatic organs, a more organized structure. We're going to look first at the lymph node, and then at tonsils and the spleen. We'll briefly look at a thymus, but it's really not functioning to filter or to kill organisms. Its specific function is in the maturation of T lymphocytes. So we'll talk about its function on Thursday. I'll talk about structure today, but we won't go into specifics about its function. So lymph nodes we first mentioned as being steps along the way that lymphatic fluid had to pass through as it made its way back to the cardiovascular system. The lymphatics are vessels. They're thin-walled, if you recall, like veins are. And so we have two of these models that illustrate locations of lymph nodes and lymphatics. And so lymphatic capillaries would pick up lymphatic fluid in the loose connective tissue or around the capillaries and carry it through lymphatic vessels. They're just identified as lymphatics. And it would enter into a lymph node. Now, a diagram on the board. It then has to filter through the lymphatic cells, the lymphocytes, and then it leaves the lymph node, passes through lymphatics to another lymph node, where it goes through that process again. So this is going to happen over and over and over and over as it makes its way from the lower extremities up the thoracic duct, or from the right side of the head, chest, and arms into the right lymphatic duct. So on this model, you can see the areas where we have high numbers of lymph nodes, and then you can see, kind of hidden back here, this is sternochyli, and entering into the thoracic duct, and then the short right lymphatic duct. So it's a huge model that doesn't show much more than you can get from pictures, but you can see those components.
Does it have a particular place it's trying to get to?
It's, remember, the fluid is trying to get back to, it's being carried by pressure force back to the blood, because we're returning that lymphatic fluid back to the blood, but we don't want to flow into the blood bacteria or viruses or metastatic tumor cells that have been in the connective tissue.
So all the nodes are pretty much just filter stations?
Yes. They're all sites for that filtration.
So they would filter, and they're going to end up in the veins.
In the veins, yes. So they're going to go into the subclavian vein just prior to it becoming brachiocephalic. So the areas where we have high numbers of lymph nodes are the cervical lymph nodes. So along, if you've been and gotten your teeth cleaned, you visited a dentist, they're going to palpate along here and see if you have any enlarged lymph nodes. You might have gotten enlarged lymph nodes in your axillary area. So if you have a sore throat, it often shows up as enlarged lymph nodes along the side. So here are the lymphatics, and then we have scattered lymph nodes, but a high accumulation in the armpit or axillary area. Where we tend to have joints, we tend to have increased numbers of lymphatic concentrations. Many, if not most, types of breast cancer occur in this area, in the tail of the mammary gland. And so their lymphatic drainage is up in toward the axillary lymph nodes. And so a radical mastectomy, they will check for those. If there's cancer present in those lymph nodes, then the breast tissue and underlying muscle tissue may often, as well as lymph nodes, may often be removed.
Very painful.
And you get the swollen arm from the lack of drainage. Obviously, a lymphatic system with a digestive tract, so all along the blood vessels, and then, as you can see with the individual in the groin area as well. Okay, looking at the structure of a lymph node, typically it's about the size of the tip of your little finger, or maybe your thumb, if you have a small thumb. And we saw some when we were looking at the cadavers for the cardiovascular system. They look like small veins, flattened veins, and they have a vein shape. So they have a convex surface and a concave surface. I'm going to diagram this on the board. The next slide provides characteristics of the lymph node. So on our upcoming lecture exam, I will list characteristics of our various lymph tissues and then ask you to identify which, kind of like I did with your exam where I had the different types of receptors and channels. Autorhythmic cells would have which of the following, or contractile cells would have which of the following. So, lymph nodes would be characterized by which of the following. Capsules, no capsules, that type of thing. Lymph nodes are the only lymphatic organ to have afferent lymphatics. So afferent or afferent is carrying lymphatic fluid to the lymph node or away from the lymph node.
Afferent?
Two.
Efferent is
So you
excellent.
can tell by the valve which direction the lymphatic fluid is going. So the afferent lymphatics are coming into the lymph node from the convex surface, and they may be coming from blind lymphatic capillaries, or they may be coming from another lymph node. If they are coming from another lymph node, at that lymph node they would have been efferent lymphatics, but as soon as they get to this lymph node, they are now afferent lymphatics. Right? So they are thin-walled vessels, that's why they have valves, and they are going to have a capsule. So lymphatic lymph nodes are fully encapsulated. Dense, irregular connective tissue. And part of that capsule will extend into the lymph node itself.
So that's going to be the dense
Pardon?
regular tissue.
Yes. So this is the capsule. And these are known as trabeculae. You may remember that term from bone, trabecular bone. Just under the capsule is where we find our lymphatic follicles. So they're going to be here, just under the capsule and between the trabeculae. And like our loose lymphatic follicles, they can have germinal centers as well. So we have the same arrangement of B cells on the T cells on the outside and B cells on the inside. So this location of the lymph nodes, the red is my own component, it's not part of the histology. But that area between the red dotted line and the capsule is known as the cortex. The rest of this area in here is the medulla. Within the medulla, we do not have lymphatic follicles. Instead, the lymphocytes form cords and divide the medulla up into channels known as sinuses. Now to our reticular fibers. Reticular fibers, remember, help to form the support for our lymphatic follicles. So we find reticular fibers at a high concentration within the lymphatic follicles and the cords. But reticular fibers are also going to be present in the spaces around the lymphatic tissue. So let me go back to the previous diagram, probably clearer than my diagram, my image. Okay, one of these days. So here's the capsule. See these little lines drawn in? Those are the reticular fibers. They would be present within the follicle as well. So when lymphatic fluid comes into the lymph node, it can travel around the follicle, that would be the easier route, or through the follicle. These spaces are known as sinuses. So the sinus deep to the capsule is known as a subcapsular sinus, and I'll write that on the board in a moment. The spaces on either side of the trabeculae are known as trabecular sinuses. The spaces within the medulla are known as medullary sinuses. We have three types of sinuses named by location. Subcapsular, then trabecular, then medullary.
It doesn't look like it's space. It looks like
Well, it's a free, along with these reticular fibers, we also have macrophages. So it allows, it helps to regulate the rate at which the fluid flows through and back up if all we had was solid cells. But still in those spaces we have macrophages and we have more loosely scattered lymphocytes. So that slows down the flow and still gives opportunity for exposure.
So subcapsular sinus,
So
trabecular
this
sinus.
space here is known as the subcapsular, not subscapular, which is what your mouth wants to say. Okay. This space here is known as trabecular sinus. And then this space in through here is known as a medullary sinus. The high dense columns of lymphocytes are known as medullary cords, and those are primarily T lymphocytes. Okay. So again, the outer periphery would be T, the inner center would be B, in boy, and the medullary cords are T. Then as the fluid percolates through, it's going to leave the capsule. And the to be oriented to allow outflow. And these are efferent lymphatics. Again, when they get to another lymph node, they'll be called afferent lymphatics. In between, they're just lymphatics.
So these ones are afferent over here.
When you say lymphatic, what part are you talking about?
A lymphatic
Things going
is
in
just
and
a
out?
vessel,
Okay.
like a vein. So the afferent lymphatic is that connecting vessel at the convex surface, and the efferent at the concave surface, yes.
Can you explain to me again where you got the B's and T's again, when you're saying the B's are in here and the T's are in there?
Okay, so let me go back to,
Please.
let me write it in here. So here's our lymphatic follicle. I'll just draw a purple line around it too. Doesn't have a capsule, but just so you can see where it is. Okay, so that's a follicle. And in the periphery of the follicle, a better color to use here. So the outer part of the follicle is going to have T lymphocytes. The center part of the follicle will be rich in B lymphocytes. Whereas our medullary cords are rich in T lymphocytes. Okay? So if we look at,
Thank you.
So if we look at this diagram again, out here where you see the solid purple would be the T lymphocytes. The germinal center is because of the B lymphocytes, and these again would be primarily T lymphocytes. So the fluid flows into the lymph node via afferent lymphatics, can flow through the subcapsular sinuses and down the trabecular sinuses, or can flow through the lymphatic follicle itself. Maybe flow is a bad word, but percolate. Then it enters, that's all the cortex. Then it enters the medulla, and again can flow through medullary sinuses or percolate through the medullary cords. Then it's done with this lymph node, and it leaves via the efferent lymphatics and goes on to the next lymph node.
So is there a difference between whether it goes through the follicles or it goes around?
It's just a higher chance of finding a B or T cell that has a receptor for its particular antigens. But then even though it's going through the sinus, it still doesn't get away scot-free because there's fewer, but still lymphocytes and macrophages in those areas.
So it's just whatever channel basically
Correct.
way is open, that's the way it's gonna flow
Yeah.
through. Right.
All right, so lymph nodes are the only lymphatic tissue or organ we talk about that has afferent lymphatics. The other ones will demonstrate efferent, but lymph nodes are the only ones that have afferent. And lymph nodes are the only structure to have, are the only organ to have medullary cords and sinuses. The thymus will have a medulla and a cortex, but it doesn't have medullary cords and sinuses. Now we're going to look at another organization of lymphatic follicles. This time it's only partially encapsulated.